All right. So today, third lecture. Very pleased to see you all turn out. Great to have people be loyal. Everybody seems to have an interest in sex. Okay. So I'm happy to keep the customers happy here. All right. So what I'm going to talk about today are sex chromosomes, because it's really interesting biology of sex chromosomes that I think a lot of people are not aware of. And this links into the study of epigenetics. Just what is epigenetics anyway, and how does it work biochemically? I'm motivated in part because, in the last OLLI year, I went to Mary Crawford's class on Beyond the Binary, and she talked about some of these phenomena, but she didn't really get into the biology very much, and I thought this would be a good complement to her. And I'm talking about kind of new things. There are some interesting new discoveries made as far as sex chromosomes are concerned. All right, so let's get into it. So basically, we know the story that among the chromosomes, we either inherit an XY if we're a typical male, or two X's, XX, if we're a typical female. And these chromosomes are different in important ways from all the other chromosomes in our body. All right, so touch on this. All right, so this is a kind of cartoon version of the chromosome. So this is the X chromosome, and this is the Y chromosome. And basically, the X chromosome is very much like all the other chromosomes in our body. It has about 800 genes on it, and those genes are not really specialized for sexual development. And of course, males and females have an X chromosome, and they need all 800 genes. If you don't have an X chromosome, completely unviable. It's just too many important things specified by that chromosome. The Y chromosome, on the other hand, is very different. You just look at it in the microscope, and it's a stubby little thing. And it's been known for a long time that it's mostly this non-coding DNA, a lot of highly repeated sequences. And its really function is not clear. Now, I took this picture off the latest edition of a genetics textbook, and it says that there are 850 genes on X, and that's about right. But it says only 50 genes on Y. And in fact, that has been changed in just the last 2 years. And I'm going to talk about what's been done there. And we know that it has twice as many genes as we used to think it does. A lot of these genes are involved in the development of sperm cells. So they are really related to male development. Now, the other thing you see that's a little odd on this is that there's these regions labeled PAR1 and PAR2, and they're the same on X and Y. So for all the other chromosomes, you've got two homologous pairs, which means the chromosomes from the mother and the father code for the same genes in the same order. In fact, the sequence is 99% identical. Well, for X and Y, that is also true for these little regions at the ends of the chromosomes. And among other things, that allows the X to pair with Y. Even though they look very different, they're exactly the same up here in the top and down here in the bottom. And that's sort of a pairing region. Now, this other part in the X chromosome, I said, we now know, have about 100 genes in it. But mostly it's a lot of just repeated and non-coding DNA. The key part is one gene right here, and it's called the SRY gene. And that's the S stands for sex determining region of the Y chromosome. That's where SRY comes from. And it is the primary determinant of sexual development in humans. So what does SRY do? So it's a kind of a small gene, and it encodes for a protein. And this protein is in this family that we call transcription factors. What that means is it's a protein that binds to genes and turns them on. But it's very specific for a certain set of genes. Now, later in today's talk, I'm going to come back to transcription factors, but the main way that we control whether a gene is on or off, whether it's being read into RNA or not, is whether it's been turned on by a transcription factor, a little protein binding the start of the gene. And that's what SRY does. So in this case, you have very early in development, all right, you have a set of cells that are going to develop into part of your sexual genitalia. If you have the SRY gene turned on, what that does is it turns on another on switch. It's a turner oner of a turner oner, if you will. And this other on switch goes to the genes that specify the development of testes in males. And once the testes start to develop, and this occurs pretty early on, week 4, 5, 6 in human development, they start producing testosterone. And the testosterone then, as a hormone, turns on the rest of male development. So it's kind of like the key switch in a whole program sequence. Now, what if you don't have that switch? What if you don't have a Y chromosome? Then what happens is you go to the default option, which is everything goes female. Or another way to put it is our cells are basically programmed to be females. And they're going to be females unless you do something otherwise, which is to turn on the development of the male organs that turns on the development of testosterone. Now, it gets a little more complicated in that it's also required in these multiple steps that you have to block in some of the cells the female development. So you don't just turn on the male development, but you block the pathway that leads to the development of ovaries. Now, I'll come back to that in a little bit. So this is a bit of a complicated diagram, and these are rather tiny, but the message here is pretty straightforward. So in humans, and I'm just going to hide this thing here. Okay, so in humans, you start out with, the female has an X and a Y, and it's duplicated them. So it's got 4 copies of the chromosome. And in the males, it's got XY. I think I just misspoke. Females have XX, males have XY. 4 copies of the chromosome. And you end up normally with, say, 4 possible eggs. In humans, when you actually do this, by the way, and you go through this division, you end up with 4 different cells. In humans and some other creatures, 3 of those cells actually never develop further. You take all of the cytoplasm and devote it to making 1 cell. And just as a little byproduct, those other cells stay associated, and in rare cases, they can get fertilized. And I can't think of the term for that. Anyway, it leads to an abnormal early fetal development, another kind of rare defect that can happen. But anyway, for our case here, what you end up normally, of course, is you have an egg that has an X from the female, and you have a sperm cell that has either an X or a Y from the male, and you end up with either an XX or an XY. Okay, that's the way it's programmed. Yes.
What it's saying, ask you to share your screen again, but I see it. I don't know why.
Yeah. Okay. So if a Zoomer is looking in there, we're monitoring it on Zoom, and it is coming through. So it's apparently not the problem's not at our end. Okay, so the other thing that can happen though, down here, for example, let's say that the woman has an egg that has a single X chromosome in it, but you had a failure to split properly from the male, and you get a sperm cell in which it has neither X or Y. And this does happen. And so you end up with the fertilized egg having X, no Y. It's an XO chromosome set. People who get that have what's called Turner's syndrome. They have no SRY gene, so the prediction is that they will be female, and in fact, they are female. All right. Now, another thing that can happen is you can get this failure in the sperm cells where the X and Y didn't separate. And the egg has a normal X, the sperm has an X and Y, and so you end up with something that's XXY. So, interesting questions. Double X, that's usually female. It's got a Y, so it's got the trigger to be male. So, what will the development be?
Male.
Male. It'll be male because it's got the gene to turn on the male pathway. All right, so Mary Crawford went through a few of these, but I'm just going to mention this again. So these are the sex chromosomes called aneuploidies, which means atypical distribution of the chromosomes. So at a rate of about 1 in 1,000, you get live births who are triple X. They turn out to be quite normal females. They are fertile, but there is some evidence, some learning issues with people who are triple X. All right, now this is 3 doses of this chromosome. You know, for chromosome 21, that's causes pretty severe abnormalities. Why doesn't it cause severe abnormalities for women? Does anybody know the answer to that? All right, I'm going to hold that back because I'm going to explain it a little bit later. This is something unique about the sex chromosomes. Now, we mentioned that you could get XXY. This occurs at a slightly higher rate, about 1 in 100. And this is male, is called Klinefelter syndrome. These people are sterile. There tends to generalize be some body shape difference. They tend to be very tall, thin people. I mean, what you have here is the main effect is this kind of, you know, most males have got 1 X, but this one has 2 X. And that extra gene dose is probably what causes some change in development. Now, what if you get an X but no Y? So this is a more rare thing, about 4 out of 10,000. This is Turner syndrome, and these women are sterile. They cannot form viable progeny, and they tend to be very short. Another abnormality you can see is XYYs. What happens if you get 2 Y chromosomes? And that turns out to not have a very large effect. And that's because, in part, because there's so few genes on the Y chromosome that apparently the extra dose doesn't really have much effect on development. And they are fertile also. And what happens if you get a Y chromosome but no X chromosome? Well, non-fertile, it makes sense. You got 850 genes in the X chromosome. You don't have those genes. You just can't develop a normal organism. All right. So, any questions on these?
Yeah, so in the case of Turner's syndrome, the source of the sterility that the woman has, is it the failure to produce eggs? Is it the inability to have a uterus? Can they have in vitro fertilization? And will that work?
Yeah. Yeah, and I don't really know the answer to that, Mark. I mean, it's a good question. I don't know. Yeah, I shouldn't make any guesses. Okay, So now there are other interesting ones. You know, this comes up in, you know, in the Olympics and you decide who's male and female. There are people, you looked at their chromosomes, they would look like they're XX. There's no Y chromosome, but they have male genitalia, male development. And you have the other side too. You've got people who are XY, and yet they, in kind of development, they're females. So how can that happen? Well, it's all about this SRY gene. So we know what the normal situation is. If you're double X, you're female. If you've got the XY, the SRY gene is going to turn development in the direction of males. But what can happen is you can get kind of breakage and rearrangement of the chromosomes such that this little piece, remember, this part can bind to this part when they pair. And then you can get, in rare cases, a breakage such that this little piece here, where the SRY gene is, can get actually fastened to the end of the X chromosome. And looking at this in a microscope, you can't tell. But now you've got this gene here that makes this protein that turns on all of the genes necessary for male development. And in fact, you end up with male genitalia, right? Even though they look like they're XX. And the opposite can also be true. What you can get is people who look like they have X and Y, and yet they're female, and pretty normal females. What's wrong there? They've got an SRY gene, but it's mutated, and it's non-functional. All right. So these are, and this in fact was one of the best data to really show the critical importance of the SRY gene. And now they can do this with mice, where you can just take this single gene and do genetic experiments in mice and show that this one gene is the trigger for going down the female development pathway or the male development pathway. Now, just
should the one on the left, the one on the right, or the one in the middle?
Here.
Yeah.
All right. So what you have, this has two X chromosomes,
I got that.
but a little piece of the Y with the SRY. And so because there's SRY, it's male. This one has kind of normal XY, but because there's a mutation in this gene here, it's female.
So both of them,
And so both of these would be female here. Yeah.
Do we know a rough rate at which we have these, so it's challenging in these cases? How does it like 1 out of 1,000 for all the mixed up things, or 1 out of 1,000?
Well, I think this particular thing is much more rare. And I'm just kind of guessing, but I think it's like a 1 in 10,000. Let's see. And then just to keep it even more complicated, there are downstream effects that can affect sexual development. So I'm emphasizing the SRY gene. But what can happen is, so normal male development pathway, SRY gene turns on, makes its protein, and its protein goes to this SOX9 gene, and that itself is a master regulator of the further downstream genes. And one thing that happens after this, that's not shown in our development and in this diagram here, is this goes and turns off some of these pathways that would normally go to ovaries. You have people who have rare mutation in the signaling gene that goes from here to turn this off. And the consequence is that male development has been triggered, and these people will have external male genitalia. Female development has not been turned off, and they will develop ovaries. They have internal female genitalia. So true intersex people, this is very rare. I think probably, again, a 1 in 10,000 kind of thing. But sort of, the bottom line, thinking about this, these are all unusual, but you think of any big suburban high school, you probably got 15, 20 kids who probably have one of these aneuploidies, or there are just so many things that can go different pathways that there's just a lot of variation in how sexual development can turn out. So questions on any of this part here? All right.
When do identical twins start?
When do,
Identical twins, when in the process does that happen?
okay, so what happens in identical twins is you get one egg, one sperm cell. So you've got one individual, and then very early on, somewhere when there are just a few cells that develop, it splits into two cell masses. In each of these, it's got to occur really early on, where you just have a few cells. Each of them then develops into a complete human being. So they're all derived from just one egg getting fertilized by one sperm cell, and that's why they're identical. They have identical DNA. So fraternal twins, on the other hand, are a case where you've got two egg cells fertilized, at the same time. And so they grow up to be two individuals. They're as different as brothers and sisters that are born, years apart. Okay.
I was wondering if this is an identical twin.
So identical twins, you have one egg gets fertilized by one sperm, but then splits into two cell masses, each of which develop into an individual. Yeah, and the splitting is really early on.
Since we're close to this, I thought I'd ask you about mosaics and chimeras.
Yeah, so actually, I'm glad you brought that up, Mark. So, I'm talking about all these chromosome swaps occur. The whole other panoply of things that can happen. There are people who say on chromosome number 6 have a tiny piece of chromosome number 3 fused to it. And I mean, I happen to know somebody in my sister's relationship that has this. And this person, girl, and she's pretty normal, but not completely. In this case, she has hearing deficiencies, slightly slower developmental problems. So it's just random what set of genes. Most of these are probably lethal. I mean, I don't know if you've heard this number. I don't think it's quite widely appreciated. But how many fertilizations are successful? You get development, but then spontaneously abort. Does anybody know the number?
50%.
It's about 30%. 30% of all successful fertilizations in vivo are aborted because they're just all of these things can go wrong.
God shouldn't be doing
And they do. Yeah.
It should have been right into the norm.
All right, so let's repeat.
So,
Okay.
I wonder, there's this thing about like the finger size and testosterone, there's all these little factors that affect things. And I wonder if there's known factors, like I can hypothesize that higher temperatures would cause, genes to split apart more. Is there any factor that tends to increase the rate of these kind of errors?
I mean, when you're talking about humans, not that I can think of. I mean, things like body temperature, et cetera. I mean, we're so well regulated. Although there's interesting phenomena like in frogs, et cetera. If you take the eggs and you unfertilized eggs and you just warm them up, give them a kind of heat shock, they will behave as if they're fertilized, and you get females developing. That's parthenogenesis that develops. So that sort of thing can happen, but in us, I'm sure there are chemicals that can mess things up, but I don't really have a good answer. All right, so other fascinating thing that happens with sex chromosomes, with the X chromosome. Why do those triple X females develop pretty much normally? But more basic question, since this gene dose, look at trisomy 21, where you just got 3 copies instead of 2, it causes quite severe developmental difficulties. Why don't females with 2 X chromosomes and males with 1 X chromosome have different levels of those genes, and why doesn't that cause a problem? Anybody know?
It's RNA to the rescue.
What's that?
I guess it's RNA to the rescue.
One of the X chromosomes is disabled in a female.
Exactly right. It's a phenomenon called X inactivation. Very early on, and you see in my slide here, somewhere after the fertilized egg is divided, you've only got about 500 to 1,000 eggs. One of the X chromosomes in females is turned off completely. Now, how does that happen? What does that mean? To greatly simplify it, that DNA is just tightly wrapped up with proteins and is condensed in such a way that the DNA is inaccessible by protein factors. And you can see this in a cell. You see a little black lump in these cells. It's called bar body, and it's just a condensed X chromosome. It is done so that you have the same gene expression level in males and females. You have a 1X in a male, and you've got an inactivated X and a functional X in females. And the biochemistry that's kind of been worked out, and I'm not like everything, it's complicated. But basically what happens is early on, there's a protein made that, and there's only enough of it to bind to a single X chromosome. And if that protein is on, it says, protect this chromosome from inactivation. So you've got a protective protein. So if you're triple X, you make enough of this to save one of your chromosomes. If you're double X, you make enough of this to save one of your chromosomes. And the other chromosome or the other 2 chromosomes are all inactivated. All right. Now, this is an interesting consequence. So this occurs at about the 500 to 100 cell stage. And so at this point, you've got, let's say, 500 cells. And the mechanism doesn't have a way of recognizing whether the X came from the father or the mother. So it's random. So in each of these 500 cells, one of the X's is turned off. Now those X's can be different. All right, one or the other. You know, the classic explanation for this, love this in genetics classes, is the calico cat. The brown gene and the black gene are on the X chromosome. You breed a black cat with a brown cat, and you can get a calico, because early on, where you had a mass of cells, maybe 50 cells that are going to be skin, they're randomly inactivated, and one of those cells that has the brown gene will keep dividing. It'll make a brown patch. But its neighbor cell that has the black gene will keep dividing, and it'll make a black patch. Now, where does this white patch come from? Well, that's just nature's way of making things complicated for us. It turns out there's another gene, a dominant white gene. So wherever this one is expressed, it's white. But wherever that one is not expressed, it could be either black or white. Now, how about human females? So one thing that's clinically known is there is a gene that affects the development of sweat glands. And you end up kind of calico for the sweat glands. So there are human females shown in my picture here, where you have this mass of cells that are going to develop into skin. And if some of them have the functioning sweat gland cells, all of the progeny of this cell will have sweat glands. But if the other have a mutant form where they don't develop sweat glands, all of its progeny, and they will develop into patches where this part of the skin might sweat, but over here it won't sweat. The top part of your leg will, but not your other leg. And so females, because of this, tend to be mosaic for some of the genes that are on the X chromosome. And this comes up for some of these sex-linked diseases. So, for example, hemophilia is the classic one, all the descendants of Queen Victoria, many of the males had hemophilia. But there are certain genes, if they're sex-linked, you can be mosaic for some kinds of diseases. Now, typically, if you've got one functional copy, it's enough. It's not expressed. But there are cases where people are mosaic for an X chromosome deficiency, and it's just kind of random chance of the draw that they have large numbers of cells with a deficiency, and they'll be partial expressors of this disease. So yeah, basically anything that you can think of, various combinations, it's really happened out there in the real world. All right. So Calico Cat. All right. So I think this is a good place to us to take our 5-minute break, which we do not want to be any longer than 10 minutes. What is the very latest on the sex chromosomes? Well, one of the interesting things is that, first sequence of the human genome was published in the year 2000, and knew from the beginning, when the initial announcement, if you read the fine print, that although it's a huge accomplishment, there was a lot missing. I figured that about 8-9% of it, in fact, had not been sequenced. But the biggest thing that was missing was the X chromosome. I mean, the Y chromosome. The Y chromosome is because it had so many repeated sequences, etc. They couldn't really line them up properly, and so it was just last year, either last year or the year before, that they finally completed the entire human genome from end to end, and the entire Y genome. And it's another Santa Cruz bragging point, because the leader of that project is a woman named Karen Miga, who's new assistant professor, young whiz bang up at UCSC. And Santa Cruz had another really key role in the ability to do that, because it was really here, a guy named Dave Deemer really started this. Dave Deemer is still around, developed something called nanopore sequencing. So he had this idea of what if we could use electrical signals to sequence DNA. Here was the idea. You'd have a membrane. This is made out of a layer of lipid molecules. And in it, you would have a protein that had a channel that allowed DNA to go through. Now, these exist because that's how bacteria can swap chromosomes. So bacteria have these proteinaceous pores, and DNA can go from one bacterium to another through these pores. Now, DNA, like most molecules, has atoms that can be positively or negatively charged. And as so as the DNA strand is popping through, it's going to be changing the current across the membrane by tiny amounts, but by detectable amounts. And so the long and short of it is they worked out a way to actually do that, to kind of have enzymes that would feed the DNA through there, and then very sensitive electrical probes. And what you would see, oops, I did hit that by mistake, is a small change in the current as each nucleotide pops through. And the real advantage of this is the previous ways of sequencing, the longest piece of DNA they could read would be maybe 1,000 bases, maybe 2,000 bases. But this nanopore sequencing can read 100,000 bases or more. And what's amazing about it is the device looks like a cell phone, God's sake. It's about this big, and you put in your little piece of DNA in solution, and it just zips through there and then get the current, and says, here's your readout. Mark,
So, I don't know if this is directly connected to the sex genes, but there has been a decline in fertility in general, all parts of the world.
mhm.
I'm wondering whether there's something here that is related to that.
Well, probably not, you know, we're just guessing on that, but among the other, you know, target things to explain it is this whole, you know, these various forever chemicals, some of which may act as hormones, you know, so that's where the suspicion is, but that really has not been nailed down.
Okay.
Yeah. Okay, so anyway, the consequence is that, so this has just come out recently. This is the complete sequence of the human Y chromosome, and this is the abstract of the paper. And as it said, that more than half of the X chromosome was missing from the reference genome that people have been working with for the last 23 years. It was the last human chromosome to be finished. The Y chromosome turns out to have 62 million base pairs of DNA. So they added 30 million base pairs that weren't there. They found in these 30 million base pairs, there were 41 additional protein coding genes, most of them in a family that's involved in sperm cell development. So quite a feat to do that. They also were able to work out the sequence and the arrangement of the repeats of all of this. This is at the centromere. And so they show this simple diagram so we can now understand it. Okay. Needless to say, I will spend no time on that whatsoever. All right. But this kind of relates to the question of epigenetics, and we're going to get into that now. All right. So, now I had, over the last 6 months, I've had a number of OLLI people come up and say, I'd like to hear something about epigenetics, and I've been wondering why. There must be something out there about epigenetics that I don't know about. Okay. In terms of the biology of it, it's interesting, but it's really complicated. You know, as a teacher, I thought that was something I almost never would say, that something was complicated. Because if you say it's complicated, then your students turn it off. Well, this one I'm not going to be able to understand. Well, this is complicated. All right, but I'm going to try to make it understandable. So what does that term mean? Well, what did that term mean to molecular biologists? And I think this is one of these many situations where a word has different meanings sometime in the popular press than it does to the people in the scientific fields that study it. So epigenetics is the study of molecules and mechanisms that can perpetuate alternative gene activity states. In other words, perpetuate through generations, whether genes are active or not. And it does this in the context of the same DNA sequence. In other words, can you somehow make the change in whether a gene is functioning or not without changing the sequence of the gene? The epi part is something on top of the gene that changes whether the gene behaves normally or abnormally. All right. And so a good example of epigenetics would be X inactivation. Here you've got a whole set of genes. You inherit them, you got them in every cell in your body. You're female, which you've turned off using epigenetics, one whole set of chromosomes, or one whole set of genes. All right. So to understand this, we've got to know, how do genes get turned on and off? And I've made some little references to this, but let's come back to this and look at it in a little bit of detail. So in the first lecture where I had a picture of a gene, and showing you how the sequence mark where the start of the gene is and where the first amino acid is and the last one, etc. So let's look at that region here in this diagram. So the gene itself, all right, would be down here, this dark blue. This is where the protein coding part is. And if we looked at where the RNA started to be copied for this gene, it would be up a little bit in front of where the protein coding region is. And it's kind of like, you know, we know the mRNA is like a piece of, you know, ticker tape or something that you feed into a machine. And this little part here is just the leader to load it into the ribosome. And then it'll find the ATG says start adding amino acids here, and we'll move on. And then in front of that, and this whole part is called the promoter, because it promotes the functioning of the gene. So in many genes, there will be something that has this short little sequence, like TATAAA, some variant of that, and that has the sensible name of, it's called the Tata box. Tata box. All right, so typical gene, you've got this control region up here in front of the protein coding region. And what will happen, let's say this is the SRY gene has produced a little protein that wants to turn on the gene for testes development. That little protein, the product of the SRY gene, may come in here and bind to this unique sequence in front of these testes promoters genes. So SRY gene encodes a transcription factor, and there are probably 1,000 or more transcription factors within your set of genes, which makes sense because there's so many things that have to be regulated and controlled on and off. So you've got whole sets that control metabolism, turn genes off and on for digesting your food. You've got whole sets for development, where you're going to turn on the neural genes or the stomach genes or the blood genes, etc. So these transcription factors come in and they provide a binding site then for the enzyme that actually copies the DNA and transcribes it and reads an RNA strand from the DNA strand. So the final step will be, this is the enzyme called polymerase. It makes the polymer. It will recognize these transcription factors. And the polymerase has within it a mechanical way to separate the two strands and then start copying one of the strands, and we'll go in this direction. So turning genes on and off really involves somehow preventing this or activating this. You make transcription factors to bind here to activate it. They mark it for, all right, this is an active gene. We provide a binding site to bring in the polymerase. Or you can make repressor proteins that will inactivate it or somehow make it inaccessible. Okay. All right. So what I'm going to show you, I did have one more video here. And what happens is normally the DNA is wrapped around these bits of proteins, these histone proteins, 2 turns, and then it goes to the next one, 2 turns of DNA. And this is 146 nucleotides goes around them. But when the gene is to be read, it's got to be open so that these other proteins can access to these sites. So what I'm going to show you in this movie here is just the kind of cartoon version of what happens. Now, what's neat about this movie is it shows you how it happens in real time. Let's see. Go. All right. And we're only going to look at part of this. All right. So here's our cartoon version of that's the DNA helix. The transcription factors bind. And then what happens is the polymerase is going to come in. And actually, the polymerase is the blue one down here. And then the whole mechanism gets turned to on, and that triggers the polymerase to take off. And this is the actual speed at which it moves down the DNA. To transcribe the typical gene with 2,000 to 3,000 bases takes about 1 minute. So you figure out it's several hundred nucleotides made per second. And so you get the RNA here with the intron produced here, and then the whole splicing apparatus comes in and finishes it off. All right. Okay. Now, so looking at, that kind of basic overview of what controls a gene, let's get to the epigenetic thing specifically. Now, part of this whole interest, I think, was triggered by this paper, which came out in 2008. Persistent epigenetic differences associated with prenatal exposure to famine in humans. And this is published in the Proceedings of the National Academy, which is a very well-reviewed journal. So this paper got a lot of attention. And it was part of a big project called the Dutch Hunger Winter Family Studies. People heard about this at all? Okay, that's interesting. So what happened here was that in the last year of World War II, the Germans put a food embargo on half of the Netherlands. And this occurred starting at the end of 1944 through the beginning of 1945. And it's important that it was the Netherlands because they're very organized people. And so even though the war was going on and there was this food embargo, the basic healthcare system continued to operate with good records, an important thing. And they also really had detailed data they can go back and look at now on food availability because they were rationing food. So they knew how much food went to what places at what time and how many women were pregnant during that period. And so it was observed afterwards, first of all, that the women who were pregnant during this period gave birth to low weight babies, not surprising, but it was found that there were certain diseases that seemed to persist in these babies for years, decades after birth. One of them, they seemed to have higher rates of certain kind of cardiovascular disease. Another one, very concerning, there's a fairly high rate of schizophrenia in these babies, which would be tied to some defect in neural development. Kind of not surprising. It's one of the most complicated things we have to develop in our bodies is our whole nervous system. So this study came out, and they were looking at these babies who were born from impregnated women in this famine period, and they were looking at them 60 years later to see if there were epigenetic changes in their DNA or on their DNA. They found the sequence of DNA didn't change, but they focused on one particular gene because they knew it was subject to this regulation. It is a gene called IFG, insulin growth factor. Now, what insulin growth factor is it is yet another transcription factor that controls a lot of metabolism and has a lot of effects on development. It has multiple effects and not simple to explain. And normally, this gene in women gets methylated. I'll show you what I mean by that in a minute. And what it does is it turns down the activity of the gene. Now, it's still not understood what the details of what's going on here. I mean, there's this complicated thing in placental mammals that you're raising this large organism within your body, and it's something you want to produce. On the other hand, you're almost competing with it for nutrients. All right, and so that's why this has to be this finely tuned regulation of how fast the fetus develops compared to how much food intake you have. And so that's why this whole factor that controls metabolism is involved. And there's doing a lot of hand waving here. I will admit this is complicated. Lots of factors involved. But when they went in to these men, 60-year-old men, they found that the degree of this methylation in this gene was abnormally low, as though it was inherited, if you will, from their mothers, from the family. All right. So here is this example. This cartoon kind of illustrates this. So you have the first generation, the mother here, with her fetus. The fetus becomes the second generation. And the interesting question is, does this epigenetic change in her apply to her children who are in the third generation? All right. So that's the question of, does epigenetics persist? All right. Well, so epigenetic mechanisms, there are multiple, but I'm going to just focus on the two major ones. How do you turn off a gene without altering its DNA? And one is you can affect the degree to which it's wrapped up in the histones, and the second, you can affect the DNA methylation. So let me go through these one by one. So methylation is a way you can silence genes. So what does that word mean? CH3, that is a methyl group. So if you have methanol, you take CH3 and then you convert it to, and you connect it to OH, you have a type of alcohol, methanol. If you have methane, CH4 is methane, all right? So methyl is just CH3. Those four atoms form a methyl group. In DNA, it is observed that often you will have this combination, two-pair combination, CG on this strand, which is CG on the other strand. And on this part of the nucleotide in the C, it gets methylated. So, I mean, that's the chemical detail of it, but the bottom line is you put like a chemical mark on the DNA. It's still a C. You haven't changed the sequence. There are mechanisms to take it off again. All right. But what it effectively does is if you have many of these methyl groups, just picture now these proteins come in to turn the gene on. Well, the surface of the chemical surface of that has been altered, and so they won't fit, they won't bind anymore. And that's why they turn off the gene. So that's one important method of epigenetics. All right. Then the second one is what happens with X chromosome inactivation. And that is what gets modified are these protein cores that are used to wrap up the DNA. So when you have this combination of DNA wrapped around this protein, the name for that is a nucleosome, and the specific proteins are called histones. You need to remember the word. But the important thing is this protein has parts on it where it can be methylated. So if you add methyl groups to this, it changes the ability of the protein to bind to the DNA. And this is a sort of cartoon version of it here. So if you have chemical modification on the tails, in some cases they can be open. So this DNA is exposed here. And if you had that Turner-Honor region in the gene exposed, all those proteins will come on, and they will actually kind of they'll push this protein out of the way when the gene is transcribed. On the other hand, if you take off this chemical modification, then the wrapping up becomes much tighter. And here, none of this DNA is exposed enough to bind those signals, to activate it. And then it becomes, you know, more tightly wrapped in that you've got what's called beads on a string. And these beads on a string can wrap up to form a thicker fiber, and then that fiber can form a thicker fiber. And that's what happens with X inactivation. The whole thing is wrapped up so tightly that those proteins just can't get to the DNA. The DNA sequence is the same, but the genes are turned off. Okay. So now I want to go back though to the the core question. So Constantine.
So what signal activates this ancient effect? So that it's thinking, I mean, what is the signaling us?
Yeah.
What does that?
Well, I mean, the way you're asking the question, could you think it? That I think is why this is so much in the literature, because people say, I am not just at the fate of my genes. I can go and I can modify them one way or the other. Yeah. And so the answer is that people have been looking very hard at the chemical mechanisms that do that. And for many of them, we understand it. So for example, for X chromosome modification, we know that there's a protein that controls the methylase that adds on the repressive signal, and the same for histones. So the answer is there are chemical signals that affect this epigenetic phenomenon. All right, but they seem, especially in humans, they seem to be especially limited to a few special circumstances. So in this case of famine, for example, you have a large number of molecules that are sensing your nutrient levels. All right. And if they get a certain level, it will trigger a stress response in your cells. So certain proteins are kind of there to protect the DNA. It's a sort of save what we have signal, where hard times are here, turn down a lot of activities, and they will trigger these methylases that will then go and trigger the DNA. It's really a multi-step process and a lot of feedbacks involved.
But I'm thinking is, for instance, in animals who have, if they need something to survive, to come here, they're thinking, oh, I shouldn't do that again or whatever. And I need this or bigger wings or whatever. That's controlled by our mind, pretty much.
Right. So this is the kind of Lamarckian idea of, can your environment actually change your genes in a way that can be inherited?
Right.
And so kind of where that stands is it can be, your environment can change things so that your body can turn genes on and off,
Right.
but they're not permanently changed. Your progeny don't inherit them. This was Lysenko's idea that if this is a real thing. He was saying, if I feed apple trees a lot of sugar, those apples will be sweeter, and the seeds from those apples will produce sweeter apples. Well, it doesn't work. All right, it's not carried out. So in this case of this particular study, all right, we could say that, all right, this mother, all right, was starving, and that nutritional deprivation signal was also sensed by her fetus, because the fetus is starving too. All right. And so it's not surprising then that when the baby grows up, these signals, off signals, epigenetic signals, they persist. But what about for her children? All right. And so in a few cases, and there's just not much evidence that this happens, there's evidence that it persisted in her children. Now, were they really inherited? Or did this third generation actually get direct exposure to the famine? Well, how could she, third generation, have direct exposure to the famine? Is your trick question for the day. There is a way.
Family.
No, what's that?
Family.
No, I'm going to say directly, her cells.
Mitochondria.
What's that?
Placenta?
Well, it's not.
The germ cells in the developing baby.
Marcus got it again. So this is the other interesting thing about our biology, is that the third generation was already here in the mother. So you may have heard this, but women produce all the eggs they have for their lifetime before they are born. And so this fetus in here, this baby, during the 9 months of pregnancy, is producing her egg cells. And so those egg cells are living in this famine environment. And so those genes could be methylated, for example, and that methylation would persist to the third generation. So people have looked hard to see whether this is carried on in humans. And, you know, I was doing a lot of reading for this to try to get up to date on it. And so far, most of the population geneticists are very skeptical. They just feel there's still not any hard evidence for this persisting in humans or any placental mammals. That kind of epigenetic inheritance does occur in plants and does occur in some other organisms. But so far, it's just not there in humans. So I think, question.
Did you define epigenetics and I missed it?
So epigenetics, again, means turning genes on and off without altering the DNA sequence. So if you methylate something, that's an epigenetic phenomenon. Or if you wrap up the DNA, that's an epigenetic phenomenon.
What about generation 4?
Well, that's what I just said. And in humans, so far, there's no evidence that it's carried into human generation 4.
So there won't be, the blue will not persist.
And part of the reason is that when eggs and sperm are made, there is basically a methylation cleaning process because during the adult development, etc., genes can get turned on and off, and you don't want all of these life experiences to be recorded permanently in your eggs. So most of this methylation is cleaned off when sperm and egg cells in the germ cells. So it's a new slate, and then the environment you're exposed to will affect the epigenetic switches on your DNA. Susan.
So what about the sperm cells? In other words, couldn't men who were born at the same time in pollen also have sperm cells that are less strong and therefore impregnate the woman with those cells?
So it looks like those cells kind of get cleaned up when the sperm cells are made. The persistent, the epigenetic chemical modification do not persist when it goes through that 4-cell division. And the other thing, the difference, the sperm cells are all made de novo. So males are not born with all the sperm cells they have. They are all made de novo. Okay.
How is epigenetics a total field for medicine? When I see that you talk about CRISPR and replacing genes, we can inactivate genes or promote genes. It seems like somebody ought to be researching a whole new class of drugs, probably less promising than CRISPR, but just another. I mean, just is that going on? Is that a big deal now or not?
Well, what specific kinds of things are you thinking about?
Well, so if you have a gene, say that you identify as a problem,
Yeah.
if you can find a way to have it wrap tighter or to occupy the sites that it goes in, you can find a drug to do that.
Oh.
You should be able to fix that. And so there should be a whole new class of drugs that basically act as epigenetic drugs.
Okay. Well, I don't know of that, and I mean, it sounds like I got some multiple steps in there where that'd be very hard to achieve, but there might be people out there. Yeah.
This methylation process occurs not just in the instance of hunger, but for lots of different genes in your body as a normal thing,
That's correct.
not just stress situation.
Right.
So, but only some of them get passed on.
Well, and so the idea is whether they get passed on through generations. And so right now it does, like in humans, it looks like they're not passed on through generations. I mean, so part of it, I mean, for example, you said it happens in lots of situations. You're right. And a good example is if you have genes specific for neural development in a blood cell, you'll turn those genes off through epigenetic means. But what happens in the generations is, of course, that those line of cells that develop eggs and sperm cells. And in those cases, all those epigenetic marks are kind of removed when the eggs and the sperm are synthesized, developed. Okay, there were a couple. All right, so what we'll do next time is we're going to actually do CRISPR, and we'll do the question of how do we change our genes? Because now we have the technology to do it. Mark?
I have one troublemaking question.
Yeah.
Why is there sex?
Why is there?
Sex.
Because it produces enormous variety. All right, because the environment changes, and if you don't have ways of swapping all the genes around and doing all these experiments, that's so part of the answer to your question was, why are there so few asexual organisms? They just die out. They don't have ways to change their genes, and they can't adapt.
So Greg, I know the sex is good. We like this. You're Greg, I know the sex is good.
Okay. All right. So hope to see you all next week for our final lecture.